Okay. So, hi everyone. So today I will present the results I had on my PhD work. I will talk today about the evaluation and characterization of rare earth elements toxicity in aquatic and terrestrial plant models. As we know, rare earth elements are naturally present in both soil and water. And actually, because of their low mobility until some years ago, they weren't considered really high toxic elements. But then, with the anthropogenic use, and as we know, us as 21st century citizens, we find now them everywhere. Their amount has increased in time. So what's really important is that rare earth elements present in soil and water also affect the ecosystem. And because among all the organisms we've mentioned these days during the presentations, plants are also a really important part of the ecosystem. And as primary producers, they play a really important role in the environment. So for my studies, I want to see the responses of two plant species, terrestrial plant species, Lens culinaris and Allium cepa, and two aquatic plant models, Myrophilium aquaticum and Lemna minor. Because the literature has shown that if we study the amount of rare earth elements present in plants, at the end we can have some comparison to the ecosystem. So it's a good way to understand in what situation the ecosystem is by actually studying the plants. Some of the endpoints that we have been seeing on the measuring on the terrestrial plant models are the reactive oxygen species production, the antioxidant system, lipid peroxidation by the content of melondialdehyde, mitotic aberration, and growth rate. While for the, in this case, for Myrophilium, which was the work that I did during my secondment in Hamburg with Professor Susan, we studied the yield of the second photosynthetic system and the growth rate. Just to make a little intro on the oxidative stress, so whenever the plant is undergoing stress that can be biotic or abiotic, we have a really high presence of free radicals. These free radicals and also our reactive oxygen species here are a subset of the free radicals containing oxygen. These free radicals are missing an electron, so they tend to attack normal cells and cause oxidative stress. And the antioxidant system comes to the rescue by protecting the whole system and the cell from these free radicals. So a way to measure the stress in the cell, an indirect way, is to measure the antioxidant system. Also, the melondialdehyde content that I mentioned before is a final product of the lipid peroxidation that happens in the membrane. So this is also another way that we can measure the toxicity and the oxidative stress in the cells. So about the work that I've done with the two plant models, onion and lentils, we saw the effect of chloride hydrate forms of cerium and neodymium in different concentrations. And in both cases, the species were first germinated in water. For lentils, the seeds were first germinated in water for 2 days, while for onion, the bulbs, the root was grown until 3 centimeters and then transferred to the hydroponic system where the treatment was made. So some of the results that we had about the growth and the mitotic aberration. Of course, with the higher concentration, we could see an inhibition in the growth of the plant. But what's really important is the lowest concentration of cerium, which showed which helped the plant to grow even more than the control. So here we see a positive effect and again the hormensis. About the mitotic aberration, as the professor already explained, they're present in all conditions. So even in normal growing conditions we have aberrations, but their percentage, in high percentage, shows also high stress. And in our species, we found these four aberrations, which are sticky chromosome, semitosis, bridge, and vagrant. And at highest concentration, 100 micromolar, their percentage was very high. About the ascorbate and dihydroascorbate content, or also known as vitamin C, this is the, in this case we measured, this is one of the antioxidant systems that I mentioned before. What's really interesting is that cerium, when treated with cerium on the root part, as we can see, it seems like the plant has no antioxidant content, while at the aerial part, at the lowest concentration, it's very much higher. And we can also see the biphasic dose response trend here. About the neodymium, the trend goes more linear with lower ascorbate content in the root and much more in the aerial part. And this is still a question for me, like why on the lowest concentration the effect seems to be more visible in the aerial part than at the highest concentration where the effect seems to stay in the root area. So about the hydrogen peroxide, which is ROS in this case, a reactive oxygen species, the high content of this element in the roots is also an indicator of the higher oxidative stress, and may be also related to the fact that there was an inhibition in the root part of the plants. The malondialdehyde content, which shows also the oxidative stress in the membrane, was significantly increased in the root at all concentration except for 25, and for aerial part at 100 for the root at the highest concentration. This was a paper that we published recently in Frontiers in Environmental Science, and like the main conclusion is that almost all the responses were activated, like the plant was having all the responses to these of course stressants, but it's really interesting the positive effect that the lower concentration had. About the research activity that we did in Hamburg, we worked with Myriophyllum aquaticum, which is an aquatic plant model that we got from the Federal Institution of Koblenz, which I really thank, and I want to thank that were very cooperative also to show us the technique that they work with. We wanted to see the effect of rare earth elements in the second photosynthetic system, if there is an effect, and how do they change in time. For this, we used artificial sediment, which was prepared by quartz sand, qualonite clay, peat, and calcium carbonate. And everything was measured by the IPAM, which normally is connected to a software in the laptop, and we put the plate and we can measure every day. The test was lasted for 10 days, and we did the measurements every 24 hours. So we also did some tests with algae, with Chantal, and we want to see the comparison between the plants and the algae. So some of the results that we had about the growth rate and the fresh weight change. As we can see, lanthanum had a really clear, of course we use the DCP as a positive control. Lanthanum showed a really evident inhibition in the growth with the growing of the concentrations, while in cerium we had the opposite effect. The plant seemed to grow even more at the highest concentrations. Neodymium didn't have a significant change, but gadolinium was really significant with the highest concentration, where at the highest concentrations plants were in necrosis, they were dehydrated, and the whey was even less than at the beginning of the test. But already at 250 milligrams per kilogram, we had signs of chlorosis, and the situation was bad since there. And then going to the yield, the lanthanum, even though had an effect on the growth, it doesn't seem to show an effect on the second photosynthetic system. While what's interesting for cerium and neodymium, on the first days of the test, they have this fluctuation of the yield, which is significant to the control. But then during the end of the test, they seem to recover from it and go to the same levels as the control. While with gadolinium, as we saw also in the growth rate, the highest concentration was really low, because of course, but what we don't know is that which is the cause of this situation. And by that I mean, was the fact that rare earth elements affected the photosynthetic system, which caused the plant not to grow? Or was it the fact that it affected the growth, and so, of course, the photosynthetic system? So, yeah, we still need to figure that out. What I'm planning to do in the future, I want to do some tests with lemna minor, the other aquatic plant model that I want to use, and we are using the sterile conditions as described in the OECD guidelines for testing of chemicals. We have been trying some growth media tests with fewer phosphates, and now when I go back, we need to also try some tests with without putting the ADTA in the medium, because with plants it's a little more complicated, as I will explain in just a little bit. We want to understand also the speciation and the bioavailability of rare earth elements, which makes my life so much complicated because I have really little background on the speciation, but the ESRs are helping me on that. We want to see also some gene expressions, and we want to do some RNA extractions on the, most importantly, the glutathione pathways, and we want to see how they are affected by the rare earth elements. The EC50, the lowest effective concentration, the no effective concentration, and as part of my secondment in Naples, I want to understand the metal uptake of the plant and of the solution that I put them in, and maybe by this experiment I will also understand my first question about how it's different, the aerial part and the root part and the aerial part, and by seeing where the metal is higher, maybe I can also give some answers to those first questions. So about what I mentioned before about the growth media, I found this work about the influence of nutrient medium composition in lemna minor, but they used it on uranium toxicity. And what they did was some tests with different media, and in my case I'm using the cis medium. But for the test to be valid, the doubling time in days should be 2.5 days. So because of that, I really can't remove phosphates at all. So with phosphate 0, the doubling time would be so long, so the test is not valid anymore. But with phosphates, this is the original, the P13.4 is the original protocol that the OECD suggests, but this is really high if I want to use the rare earth elements. So the P3.34 is the one I'm using now, and the lemna is growing it's valid. The tests are it's valid because the doubling time is within the range. But I need to try also if the doubling time will stay the same if I remove the EDTA, and with that also the chelated iron. Yeah, I need to make some trials on that. So that was it. Thank you for your attention.
Thank you, Isidora. I don't know if we have some questions. Go ahead, Antonia.
So you intending to test more in the future?
I don't know, maybe, but I really want to keep at least these four, so at least I can have a comparison between species. So if I use the same rare earth elements, I can compare better the species and their responses to it. So for now, let's keep it four. I don't know.
According to the results you have until now, which part of the plant would you say is the most sensitive?
So yeah, about the until now, I would say the root, and it makes sense because it's the part that is also immersed directly in the rare earth element in the solution. But what I'm interested to know is how they travel in the plant. At least at the lens culinaris, I can do the trial. Because at the lowest concentration, 5 and 25, they tend to have the highest effect in the aerial part. So I'm thinking then the rare earth elements is going up there. But on the highest concentrations, already the root stops growing. The effect is really concentrated in the root. So I want to see maybe by another protocol with the fluorescence to see how the plant travels. But I don't know about that yet, but I'm planning to, if I have time.
Some techniques regarding some imaging like what I did, but where you can also analyze them over time with laser ablation?
Yes, there is a way that I can, like with fluorescence microscopy, I don't know the protocol exactly to explain it now, but I can see how the plant travels how the metal travels. But and even without that, if I have at the end the uptake of each part, I can still get a feedback out of that. Yes.
Sorry, I had a question out of curiosity because we have some similarities in the project. So how did you conclude that the phosphates had an interactive effect with the toxicity of the rare earth elements? Was that literature or was that?
Yes, and I've, from all the presentations we've also had before, I've always heard phosphates affect the toxicity of rare earth elements. So before starting my trials with rare earth elements, I wanted to make sure first to use the least phosphates possible, and now the least, or maybe not at all, ADTA.
But so then are you talking about the precipitation of rare earth elements with phosphates, or are you talking about actual toxicity mechanisms within the plant?
I'm talking also about the precipitation and maybe the bioavailability getting lower. So if I have a high amount of phosphates, I risk to have precipitations, and I need to treat the plant with really high concentrations to see an effect. But these concentrations aren't real at the end because I have precipitation. So this is what I'm trying to, but I'm still in the, this is what I'm trying to understand.
Okay. So do you think then that's also a concern, I don't know, like with algae you need phosphate for them to grow. Do you think that with the plants it would also be a concern that if the rare elements form complexes with phosphate, there's less phosphate available for the plant?
I'm really scared of that. Like I want to open the freaks and do the calculations, but I'm trying, as I said, to keep at least the control in the optimal conditions, because of course then I couldn't compare. And I want to make sure that creating the best media for the plant to grow and to have the least precipitation possible and the highest bioavailability with the least rare earth element that I put in, if it makes sense.
Because you
Sorry.
used the,
No, go,
you
sorry.
used this time media, right?
I use the
one you used here in the HAW.
It's not the same, because with the myrophilium I put it in the sediment, while in this case the lamina floats, so it's only media.
Sorry, but regarding the idea of speciation too, a lot of times we're looking at what's thermodynamically favored. We don't know how long it actually takes for that to happen too. So you putting phosphates doesn't necessarily mean that they will precipitate out unless you do tests for anions to see if it is actually precipitating out, or if you do measures of the metals before and after.
Okay.
Sorry, that's just a thought.
No,
It's
yeah,
not
I
the
really-
same idea. And I actually saw that my phosphates were still in solution.
Okay. No, and I really need feedback on that because, as I said, I really don't have background on the speciation part. But also from all of you guys, I know now and from literature that also another fact is that lamina itself changes because you already have changes with the media, and if you put rare earth elements in there, already changes. But then if you put the organism in there, it's even worse because the pH changes, and with that everything else. But yeah, maybe I'll ask you again about the ideas that you have. Thank you.
Do we have maybe one last question? No, you're all set. Well, thank you very much.
Thank
Isidora.
you.